Hello everybody, welcome to the Dr. Harris channel. Today we're going to be looking at the insulin signaling pathway, which is very important to understanding how cells register diabetes, how it plays out for the cells. We went over the vocab, I gave you the outline, so we're ready. If there are any questions concerning the video I have gotten for you, ask. If there are comments, put them down and I will address them as I always do. I don't feel there's anything I need to preface to do for you guys. I think this is going to be exactly what we need in terms of understanding insulin from the moment you ingest food straight into the stomach, going through the digestive tract, how it gets utilized, what happens at the cell membrane. All of that stuff is going to be outlined quite nicely, so repeat as often as you need to. All right, I'm ready to get you in, so let's go.
When we ingest food, it travels along the digestive tract, where it is broken down into its component nutrients. One such nutrient is glucose, which is a simple sugar. It is absorbed by the stomach and intestine. Once glucose is absorbed into the bloodstream, it increases the blood sugar level. As a result, it sends signals to the beta cells of the pancreas to secrete the hormone insulin. Insulin is a very important hormone in our body that regulates the blood glucose level. These glucose molecules circulate through the bloodstream and enter into different cell types, such as muscle cells, fat cells, and liver cells. In fat cells, glucose is used for triglyceride production. It acts as an important energy reserve molecule. Now, we will focus on insulin signaling transduction pathway. Here is a fat cell. This is the extracellular space. This is the intracellular space. The insulin signaling transduction pathway consists of a cascade of events, which will lead to a series of outcomes. First, insulin binds to the insulin receptor, which consists of two identical chains. Each of these chains consists of two individual units. They are alpha unit and beta unit. Insulin is the primary messenger of this particular pathway. It binds to the alpha unit of the receptor. It causes the conformational changes of the beta unit. As a result, the beta unit is phosphorylated with presence of ATP, adenosine triphosphate. And then it activates the insulin receptor. IRS1, insulin receptor substrate 1, which is a protein, attaches to the phosphorylated insulin receptor. Insulin receptor substrate molecules are called adapter proteins, because, instead of activating anything, they actually act as attachment points for other proteins. Once it is attached, the insulin receptor substrate itself is phosphorylated into 4 tyrosine residues. Then, it provides an attachment point for PI3 kinase or phosphoinositide 3 kinase. Once that attachment takes place, it causes the active site of phosphoinositide 3 kinase to move inside of cell membrane. After that, it causes phosphorylation of a specific type of a fat molecule, which is situated in the cell membrane, known as PIP2, phosphatidylenoacetyl 4,5-bisphosphate. PI3 kinase takes a phosphate group from an ATP molecule. After that, It replaces that phosphate group on the PIP2 and creates PIP3, or phosphatidylenoacetyl 3,4,5 trisphosphate. PIP3 moves along the cell membrane and eventually ends up on a protein known as PIP3-dependent protein kinase, or PDK1. Once it binds to this protein kinase, it is able to activate an important protein kinase, known as protein kinase B. It is also known as AKT. After that, AKT goes towards GLUT4 vesicles, which are glucose transporters. Once AKT activates the GLUT4 vesicles, by inhibiting Rab protein, GLUT4 vesicles can freely merge with the cell membrane. When there is no insulin, Rab proteins inhibit the translocation of GLUT4 vesicles to the cell membrane. Once the GLUT4 vesicles are merged, a large quantity of glucose can move into the cell. Once glucose is inside, it can be utilized for ATP production, or can be stored in the form of glycogen. It is the signaling pathway that ensures only the correct molecules will be allowed to enter the targeted cells. This insulin signaling transduction pathway is necessary to keep blood glucose levels stable in the body. If pancreas is not producing enough insulin, the insulin receptors in the cell membrane are not stimulated. So, the proteins inside the cell are not stimulated. GLUT4 vesicles will also not be activated. Instead, GLUT4 will stay inside the cell and not move to the surface of the cell membrane. So, glucose molecules cannot pass through the cell membrane to enter inside the cell. As a result, blood glucose level increases. This is called diabetes mellitus type 1. If pancreas produces enough insulin, but the receptors are not working properly, this is known as insulin resistance. This can occur when the cells, the receptors, or the proteins are not working properly. So, glucose molecules cannot pass through the cell membrane to enter inside the cell. It is known as diabetes mellitus type 2.
Alright, so I hope you enjoyed and learned from the video I have had designed for us in regards to insulin and how it is in the body, how it responds to sugar, the role insulin plays, the role of your insulin receptors, how diabetes type 1 is presented to the cell, how diabetes type 2 is presented in the cell, and the definitions. Now, I went over the definition of diabetes type 1 and type 2 in our outline video, and again, I had the video with the cartoons say again what diabetes 1 and 2 is for the cell. So that's where we're going to end. Again, questions, put them down. I'll address them. Share this video with everyone that you know that needs this information. Subscribe to the channel if you haven't already. Thank you so much for viewing the Dr. Harris channel, and I will see you soon. Our next video is going to be on the types of bioavailable products that we have that can help our body to do better with sugar maintenance. That's going to be the follow-up video to this. If there aren't a lot of questions or any questions in regards to the insulin pathway and our introduction to diabetes based on the cell. So with that said, have a good day, be well, and I'll see you soon.